Hello there everyone, welcome back to Ants Authority. Today we will discuss about the new study that shows the social isolation shortens the lifespan of ants. In this video, we will discuss about this topic in detail. So let's start the video. On 27 September 2023, researchers from the University of Lausanne and the National Institute of Advanced Industrial Science and Technology conducted research to determine the impact of isolation on ants' lifespan and published results in Nature Communications. They found that oxidative stress, hyperactivity, and disturbed sleep are responsible for reduced lifespan in socially isolated ants. They engage in group activities and coordinate to ensure survival due to their social nature. They cannot live as a single entity and die before the completion of their lifespan when they get lost in the environment accidentally. Some researchers conducted research to determine ants' behavioral changes in isolation and found a reduction in their lifespan. Social insects adapt to survive in the form of groups and engage in collective activities because they are dependent on each other, while solitary ones can live alone in solitary conditions. Isolation is not a problematic thing for solitary creatures like ladybugs and praying mantis, but it poses severe stress on social insects that are not adapted to survive alone. It has a negative impact on their behavior and lifespan, and such changes lead to the early death of ants, honeybees, and other insects that make colonies and live together. Moreover, they perform specific tasks in the colony for their entire life and depend on fellows for survival. So they cannot survive aimlessly and die quickly when struggling with loneliness. Akiko Koto, Thomas Key, Laurent Keller, Mukato Temura, and a few other researchers from the National Institute of Advanced Industrial Science and Technology, located in Japan, and the University of Lausanne, Switzerland, conducted research to study the impact of social isolation on the lifespan of ants. They used carpenter ants as a model organism and studied genetic or behavioral changes during isolation, which were collected from queen right colonies. They had the same genetic background, age, size, and were kept in the same environmental conditions to identify the behavioral changes according to their number. A group of 10 ants were placed in one box, and an individual ant was kept in another plastic box of the same size. Honey, water, light, and eggs were provided to create a suitable habitat. They extracted RNA from ants living alone and in groups and used it to synthesize cDNA to sequence the bases with an Illumina high-sec machine. They performed RNA sequencing and analyzed transcriptome level differences to compare the changes in socially isolated carpenter ants with those living in groups. Analysis of the phylogeny of CYP clan 3 differential gene expression of cytochrome P450 and ontology through advanced technologies help them to find changes in gene expression. Also, they tracked movement by capturing photos of their movements in the plastic containers and found the orientation of their movement. They compared their speed, sleeping patterns, metabolic functions, immunity, and eating behavior from those living in groups and found significant changes. Thanks for watching the video.